Hello and welcome to the Masters of Beautiful Achievements podcast, a podcast about innovative solutions and better business models inspired by natural science, systems thinking and leadership. My name is Alexander Prinsen and great you took time to listen to this new episode. These conversations are made possible by our patron community. Thanks to your monthly support, you are helping me to record these inspiring conversations. So thank you for your support. When you're not a member yet, click on the link below this podcast to join and help me continue. Looking for more inspiring episodes, check out my website mastersofbeautifullachievements.com. You can also find the links here below. This episode is part of a special series where we look at microbes through a completely different lens. Mark van Bemmel from Orvion and Alexander Prinsen from Scope Matters explore how life sciences, biotechnology and environmental sciences are influencing how we look at our natural world. What happens when we zoom out and look at microbes as important players of a much broader system? So together with specialists in the field of virology, microbiology and biochemistry, we go on a journey combining the insights from the various disciplines to learn about the essential questions we might have been overlooking. And who knows, we might discover one or two innovative applications along the way. Our sponsors for this episode are Scope Matters, a systemic innovation agency from Rotterdam, the Netherlands. They accelerate businesses through systemic innovation by merging ecological thinking with economic doing. Their systems approach transforms and improves the business model of their clients by creating new opportunities, mitigating business risks, and create new cash flows, while at the same time eliminate waste and do good. It is a space where systemic sustainable thinking meets circular innovation, because in the end, all matter counts. Check out their website on scopematters.com. Orvion, a young biotech company from Stolwijk, the Netherlands. They develop microbiological solutions to remove contaminants from soil, water, or even industrial processes more cheaply and permanently. Their services are available for water treatment, soil remediation, ecology, and industry. Orvion believes nature can do a much better job, so please take a look at their website orvion.nl. And now it's time to enjoy the conversation with our masters. So in today's conversation, we will explore how we can learn more about the world of microbial ecosystems and how they can help us design better biotech solutions. My name is Alexander Prinsen, and together with my co-host Mark van Bemmel, we'll explore the fascinating world of microbiology. Fermentation used to be done using single microbial species, and yet this is not how nature does it. So we decided to ask a microbial expert to join our conversation about multicultural fermentation. And we are very delighted to have Diana Machado de Sousa from Wageningen University to join us to explore this topic. Welcome, Diana, and it is an honor to have you on our podcast.
Thank you very much, and good afternoon. It's a pleasure to be here.
Welcome. Welcome. You're from Portugal, and how do you end up from Portugal to the Netherlands?
Yeah, well, that's a good question. I never planned it. It happened a little bit. So it's of course related to my professional career and the way everything developed. So I studied in Portugal biotechnology. So that's biological engineering was my basis study. And then I started a PhD, still very much focused on the biotechnological conversion, on the biological conversion of lipids. And it was looking into the process, but we also wanted to look more into the microbiology. So these were microbial conversions, and we wanted to look more into microbes. And to do that, and I had to come to Wageningen during my PhD because here there was a long tradition already on the anaerobic conversion of organic compounds. So that's how I first came to Wageningen, and also where I really got fascinated by microbiology, and I switched a little bit my career route into the microbiology. And it was very important in shaping, I think everything like from the beginning till now, it makes sense how my career developed in the way that I started with biotechnological applications. I like that because it's to do with a better future society in which we can close cycles and have more a more sustainable society. And then I like natural solutions, so microbes can be used for natural solutions. And another thing I like is team playing. So I like to work with other people. And in that sense, I also like to work with different types of microbes together. So I like to make teams of microbes. And during my PhD here, I was working with these sludges with many microbes together. And specifically for the conversion of these lipids that I was studying, you need at least 2 types of microbes working together. Otherwise, it's not possible to degrade them anaerobically. So you need these organisms called syntrophs, and syntrophy means literally like eating together. So they need to be together to convert the lipids. So that was really nice that I could already then really explore how microbes can play together and can do different or more powerful things if they are together. And this reflects even now in my research, because the way I'm now doing research is also, well, the question now it's not lipids anymore. I'm much more focused, for example, in the conversion of Syngas, that maybe you want to ask later what this is, but it's well, a gas that can be used by microbes, and there are already processes for the conversion of this syngas by pure cultures. But what we do is to convert this syngas by using teams of microbes. So that's still
that's new.
Yeah.
Yeah, because the second conversion is Lanzatech, isn't it?
So there's Lanzatech has started many years ago with the conversion of carbon monoxide, especially from steel mill industries, because they, well, one of the flue gas components in the steel mill industries is carbon monoxide. So that's a concentrated stream of this compound, and they produce ethanol from this steel mill industry. So CO is the main compound, and CO is also the main compound in syngas. But when I talk of syngas, my goal would be not to use the steel mill CO, but more using syngas that is generated from waste or from biomass, so that we can have what we call a biosyngas, so a syngas that is produced from a renewable source.
Yeah.
So we can have, because you can have like biomass from trees or a bit recalcitrant waste. And there's developments on gasification. So gasification is the process where these carbon-containing compounds are burned, but they are burned with low amounts of oxygen. So then you don't have the complete oxidation of the material to CO2, and you form CO. And this CO then can be used by microbes. So we can construct a very, well, circular type of economy if we have wastes, gasify them, and then convert them with microbes to chemicals that are needed in chemical industry, and that at the present we are still obtaining from petrol. So it's a future alternative, I hope.
Why is syngas so important as a building block or something like that? Or?
Well, syngas, it's a very energetic gas because it has CO, it has hydrogen. So these are high energy compounds.
Yes.
And they are also used, they can also be, there's many chemical processes also to convert these gases to different types of molecules. The thing with microbes is that they can take a little bit higher concentrations of what is called impurities that also are present in this syngas. So this would be one opportunity for microbiology. But why is it important is you can take sugar, or you can take, easily degradable corn, for example. So all these first generation feedstocks, you can take them directly into microbiological processes. But if you think of more difficult substrates to use, like trees that have at least 20% lignin, that it's very difficult to convert. Or if you take waste that is mixed and recalcitrant, so those are very difficult to take into a microbiological process to valorize.
Yes.
So if you burn them, then you get always the same type of substrate that you can use. So it's a little bit, if you want to use an analogy, it's a bit like constructing, remodeling a house or constructing a new house. So if you take the substrate as it is, and then you try to make molecules, you are trying to reconstruct the house.
Yeah.
And what we do is to completely destroy the house and then build a new house from scratch.
So you take down the wall, but use the bricks again, or something like that.
Yeah, exactly.
Right. So, and then CO is the
brick.
the brick. Mm-hmm. Yeah, okay, I can imagine.
But you need to put a lot of energy in there first.
The gasification, that's totally not my expertise because that's really a chemical process. But indeed, to initiate the reaction, you have to put energy, but then it also produces. It's also exothermic reaction. So I think then it keeps by itself. So it's a feasible process that you can use. And of course, there's many research also there and many developments, because traditionally gasification was also done to coal. So people were doing this to obtain syngas from coal as well. So the research has been now in the last years to apply this process to biomass, or even to high or to substrates that contain more humidity, because that's one of the problems also of gasification. If you have to heat up a substrate that has a lot of water, then it costs more energy.
So then we have CO as the carbon building block, and then we have H2, which is mainly the energy, probably.
Also, CO is energy.
CO itself also. Mm-hmm. And then you can build large molecules again. Is that right? Yeah.
Yeah, you can make so the anaerobic microbes that have the capacity to convert CO. So they basically take 2 molecules of CO to produce 1 molecule of acetyl-CoA. So acetyl-CoA is a 2-carbon molecule that is a very central metabolite in the microbes, from which many other molecules can then be produced. So indeed, you increase the length of the molecules that you are produced.
Right.
So CO is actually used for carbon, but also for energy, because they take 1 molecule of CO and then they also oxidize part of this CO to CO2 to generate reducing equivalents, and then they take these reducing equivalents also to reduce CO2.
And what kind of interesting things can you make then from that with your bacteria?
Well, natively, so the microbes that are known that use carbon monoxide for growing, the anaerobic ones, they can produce, for example, you have the ones that produce hydrogen. Mm-hmm. You have the ones that produce methane, so methanogens. They can also use carbon monoxide, some of them. Mm-hmm. Or you have the ones that mainly, the acetogenic organisms, they mainly produce acetate.
Acetate, right.
And some of these ones, they are also able to do solventogenesis, then they also produce ethanol. And then in lower amounts, some of them produce, for example, butanol, or even in lower amounts, hexanol. But the main, I would say, like, the most of the research on syngas conversion presently is with acetogens. So it's the ones producing acetate and ethanol. And they cannot gain a lot of energy from this conversion. It's not in terms of ATP, and I don't want to be maybe too technical, but in terms of ATP, they cannot make a lot of ATP from CO conversions. So you cannot easily engineer them to produce compounds that, genetically engineering them to produce compounds that demand a lot of energy, because they just don't have it from this molecule. Mm-hmm. And that's why it's a little bit limit if you want to go into genetic engineering of these microbes and to produce very complicated molecules, then it gets a little bit difficult. And that's also the reason why we then thought, okay, if we cannot really change this one, why not coupling another organism that takes up the products that are produced by these carboxylotrophs, so the one that can convert CO, and just
but do you need to
be inventive and you can plug in whatever microbe you want, or 1 or 2 or 3, and then create different food networks and produce different compounds.
Yeah, so
But then you need to add electricity to it, or protons. What extra energy source do you need to add, or are other microbes specialized in producing more energy for those who find it who need more than general?
So these acetogens, they get both the electrons and the carbon from the CO. So the electrons, they get it from oxidizing CO to CO2 and one couple of the electrons. And they also are able to integrate CO or CO2 in this pathway, the reductive acetyl-CoA pathway, and then produce acetyl-CoA that can also be used for growth. So they are getting from the same compounds, energy and carbon.
Right.
But it's a good question about electricity, because I already said they have to oxidize part of the CO, so they can fix to CO2s or to C1 in this pathway. Yeah. But if you give them only CO, they will need to sacrifice part of this CO to generate reducing equivalents for the pathway, right? So there's always a production of CO2. So we are still losing some of the carbon of syngas in CO2. And you could also think like there's hydrogen, so this hydrogen can be used to fix then this extra CO2, but the hydrogen in the syngas is not sufficient to fix all the CO2 that is released. So you could think of adding extra reducing equivalents to the system in form of additional hydrogen or in form of electricity to completely fix the CO2. So that's a good remark, yeah.
So actually, you are producing CO2 in this process. Is that right?
Depends how you.
If you use
depends how you look at it.
Okay.
Yes, you still produce if you have CO or if you have syngas, because of what we were discussing. There's not enough reducing equivalent to fix all the carbon, so we still have some CO2 that is lost. Yes. If we don't provide the extra reducing equivalent. Yeah. But we are still fixing more than if we would not do anything. So yeah, it's but it's and so we are also working on solutions to fix this CO2 by hydrogen supplementation or electricity supplementation.
Are there no bacteria who are able to consume CO2?
Yeah, also these most of the organisms that use CO, they are also able to use CO2. And the thing is that for CO2, it's a completely oxidized molecule, and they still need the energy, the reducing equivalents coming from somewhere. So that's the hydrogen or the electricity.
More hydrogen in that case.
Yeah.
So plants use photons as an energy source to break down CO2, and that is a huge energy source which bacteria do not have at their disposal.
Bacteria are very, very flexible, so I would not dare to say that there's anything that bacteria cannot do. Now, there's also bacteria that can use light, and also carboxylotrophs. So carboxylotrophs are organisms that use carbon monoxide. So there are also organisms that use carbon monoxide that can gain energy from light. For example, Hodospirillum rubrum is an organism we work with as well, and it can use energy from light. So that would be yet a third option to get energy for these carboxylotrophs. Of course, the question there is if it's easy to get these organisms exposed to enough light in a process. So that's the same questions you get also with, of course, algae or photosynthetic.
On the other hand, if you're using gas fermentation, I could imagine that you just blow in some extra photons. In a way, I mean, it's still bubbly.
You need a glass reactor then.
Yeah.
Yeah, I mean, I have no idea. I mean, light can travel through many ways.
Oh, you have these reactors for growing algae. They are made of glass or of transparent, how do you say, material. So yeah, you could add photons to a bioreactor. Of course, it's possible. Yeah. But it's not that easy, by the way, I think, because your your liquids are quite murky, so your light doesn't travel too far, probably.
True.
Yeah.
True. But that's
So you work with bioreactors then, Diana?
Yeah, we do work with bioreactors, but I don't know if the type of bioreactors that you imagine. So small bioreactors, we are always in 1 liter scale. That's our type of scale.
And then your bacteria, you must use at least two different species. You told us.
For this synthetic co-cultivation, yes, at least two. And we try to increase the complexity, but the more complexity, the more complicated it gets. And also the more surprises you get. And that's actually a very nice thing when we started this line on co-cultivation. You, of course, envision which microbes you want to put together, and then you envision also how they will exchange metabolites, or you have in your mind, okay, this one will make this conversion, and then the second one will make the other conversion. And that normally still happens like that, but there's many small things. If you look into more detail on what is happening in the culture, actually, then you find many other things like amino acids interchanges, or, we also saw with transcriptomics that you start having more flagella genes being upregulated. So all these small things that are probably related to microbial interaction start popping up, and there's always something different. So that's also, they are quite exciting to see.
So how does this work in practice? You have an idea, you want to make something, let's say ethanol or something like that, and then you just think, well, let's put together this acetogen or this syntrophic organism. It should perhaps it works. Is it something like that?
Yeah, I think that's a little bit how it started, but ideally, I think we should move to a different. So I've been discussing also with colleagues more on synthetic biology and modeling, and we also collaborate with groups doing the genomic models of how these interactions go. So ideally, I think if we develop further this field, we should go to a situation in which we can predict a lot of this already from a model or an in silico approach, that we can construct databases of, or there are already some databases on physiological data, so that we can mine this data, and that we can also ask the help of computers to make pairs of microbes. Well, there's software for pairing a lot of things, so you could also have softwares for making the right pairs and the right teams of microbes. So I think that's how it should be developing.
Because if you just choose two species, they won't easily collaborate, is it? They have to.
When you know a lot about them, maybe it's like making couples. If you know a lot about them, then you kind of think it will work, but indeed, you are never 100% sure. Right. So we were quite lucky with the couples we made in the lab. They were very happy together. Yes, I think there should be more the development towards algorithms that can help the selection of these microbes. But that's, I think, where we are going. And I think we need to still do a lot on good characterization of microbes, of single microbes, because even if you have, if you need to provide all this data to computers to have the models reliable. So we need to know about individual microbes and to organize all the information, because there's also a lot of information, but scattered or in paper. So I think there's the need to really compile all the physiological information in a very comprehensive database. And there's some examples, but I think we can always improve there. And then really try to set what do you want to set as basis for a good collaboration. And this could be conditions. For example, pH is a very important parameter for the functioning of these co-cultures, because you cannot have an organism that likes to grow a pH 10 together with one that likes to grow a pH 5. That will not work. So you have to define the borders and then ask the models, like, what's the ideal collaboration for this.
Should this work? Yeah. So, and then you have these two organisms in your reactor, and then you try to persuade them to do something nice. But then how do these two organisms, how do they collaborate? Can they communicate, or?
Yeah, those are the questions we still have also, because as I was saying, when we put them together, there's always something that they do that we did not expect before.
Exactly.
So we go, and it's nice that now we have so many tools that we can look into more functional omics as well, like transcriptomics or proteomics. So we do use these tools to learn more about other ways they communicate.
Right.
And this involves, for even, quorum sensing. You probably heard about that, or molecules that they also, so there might be molecules they exchange, or even that they release to the medium to attract each other. So this type of microbial talk, let's say, that we don't know so much about.
Mark van Loosdrecht told us something about that. They can throw out a molecule and somebody else picks it up, and
and then they create a biofilm. That was that's what you mentioned.
Biofilms, that's very important. So we do also see. So I work with these synthetic cultures, but also with still with the natural syntrophs. So the ones that occur also in nature together. So for the synthetic cultures, we don't care if they come from different environments. We just care about what they do, and then we think, okay, they do this and they match with these other ones, so we put them together. But maybe they were from completely different environments. The first co-culture that we did was one organism was isolated from rabbit feces, and the other organism was isolated from the canal in Delft. So they were from completely different environments and not often reported to be together. So that's really based on the metabolism, but we still work also with the natural syntrophs, because I think we can learn a lot from them, from those. They naturally evolved to be together, so they should be able to tell us something on how to collaborate. If they were collaborating for millions and millions of years, I think they have something imprinted in their DNA on how to have good collaboration.
How to do this, yes. And how do you think they do it? Do they
that's
to each other, or how do they?
Of course, they need to be together to do certain conversions, because alone, if for example, for butyrate conversion, and if you have no external electroreceptors in the medium, and I'm talking about sulfate or nitrate or oxygen, so if you have a completely anaerobic environment without these compounds, like it happens in aerobic digestion or in the soil, in the sediments, in many environments. So butyrate oxidation to acetate and hydrogen is endergonic process. So it demands energy. It's not a reaction that naturally occurs. It will only occur if hydrogen level, so one of the products from that reaction, if hydrogen level is kept very, very low, then that changes the thermodynamics and the reaction is feasible. So there's this organism that is doing the oxidation of butyrate to acetate and hydrogen, and then there's another organism, the methanogen, that takes the hydrogen. So it's always very, very low in the medium. So together, if you sum the thermodynamics of both reactions, it's a favorable reaction. So they need to be together, and that's also what, of course, keeps them together.
And then
they also need to
really
find
stick together?
They also need to find, they need to find each other. So now we are looking, we have some projects now also on looking to these organisms and also looking how they actually stick together. Because if you look to anaerobic digesters, many times there's these granules. Well, you know about that, right? Because you studied in Wageningen and environmental technology.
Maybe Alexander doesn't.
No,
you should explain a little bit about these granules.
Yeah. So if you have if you have anaerobic digestion has been used to convert many different types of effluents from industry containing lots of organic materials, and it was discovered by in the group of Lettinga in Wageningen that if you apply a very fast flow through a reactor, you create a lot of shear stress. So you have like a reactor, and the liquid feed of that reactor is going at the bottom, and the exit of that reactor at the top. So you have you have an upflow flux And microbes are very small, so of course they will be dragged out of the reactor. So the strategy for them to stay in the reactor is to aggregate and to form
To make
small,
themselves heavy.
Yeah.
Yes.
So they make them exactly. So they make themselves heavy by forming these granules that you can see. They are like 4 or 5
Oh wow.
millimeters.
You can see them, yeah.
So then they stay in the reactor.
And these granules are only comprised of bacteria.
Yeah, basically. Well, also a bit of EPS, but this EPS is also produced by the bacteria. So most of the things are
It's like a snowball, but then a snowball purely from bacteria.
Yes, like that.
Oh, wow.
They stick together and then they can stay in the reactor.
Yeah.
Because of their weight. Yeah.
I would imagine that they grab something like organic mass and then build around it, but they have enough weight together. Wow. Okay, cool. It's like a bacterial football.
Yeah.
The bacterial snooker ball. Okay.
Yeah.
And you see,
and these granules. So these granules in these reactors, it's thought that this the pressure, the liquid flow, it's a driver for this because they really need to get bigger. So it's probably even the bigger thing. But we also see now in our co-cultures, and we are studying that more in detail of these syntrophs, that they start aggregating. Just the syntroph and the methanogen, and that's because they need to be close to really exchange very well this hydrogen between each other. And another thing we are looking then, it's like, how do they get close? So what kind of, are they excreting any kind of substance that attracts the two species together? Or so that's,
do they swim to each other? Or?
Yeah, how do they sense that there's another partner there and that they need to be together?
Yeah. You have any ideas already how this works? Or?
I would not dare
speculate. We're not scientifically reviewing you.
Of course, it's still too.
I
guess too difficult to say. There's in bacteria, in aerobic bacteria, there's a lot of studies done on coron sensing. These are these signaling molecules that microbes can excrete, and they are a signal to the other ones. So one thing we are looking is at this. So we are searching for the common signaling molecules that are also found in other microbes. But there might be also other. As I said, these are mainly studies in aerobic organisms. There might be also other molecules that are specific for these for these microbes that we don't know yet, that have a role as a signaling molecule. So it's a little bit still swimming in the dark. But
then still, you have one bacteria here in your reactor, and then the other one here, and they sense that they are there. They sense each other, but then how do they get together?
Well, they can, some of them, they can, like the syntrophs, most of them, they have flagella, so they can swim.
Right. So that's what happens. They will swim towards where the compound or the signaling stuff is coming from. Is that right? It's amazing.
It's interesting because I know a story about seaweed, and seaweed is able to disrupt the communication between bacteria. That's why seaweed has less problems being eaten by fungi or bacteria, because they're able to disturb the communication, which is in a way that they can disturb the peptide molecules. So
you don't need those in your reactor.
Okay, no, I did not know that, but.
And I don't know, I can imagine it's sort of like they make a string out of all these peptides, and then once a bacteria finds one, and then they just follow the string and then find the source of the excretion. I have no idea, but I can.
Yeah, well, I did not know that, but maybe that's a good idea, just to add these compounds to these cultures and see if you disturb the
run away.
granulation and the communication between the organisms.
One other question I found from your research, Diana, is also something about sulfur and dimethylsulfide, the sulfur-reducing bacteria. Why is sulfur? I mean, there are bacteria which can digest sulfide, isn't it? Or is it a source?
Yeah, sulfide is completely reduced, so they can oxidize sulfide,
Yeah.
but then you need an oxidizing.
But
oxidant.
how do they oxidize sulfur? They
can convert it. Yeah,
they
with
take an
oxygen,
oxygen somewhere else and then.
Yeah.
Oh wow. Okay.
Yes.
But that's not something that we are researching, so we don't work so much in the oxidation mainly in anaerobic environments, you would have then the reduction of sulfate to sulfide. So not the reverse. It's not so much what we do. Yeah. But we do work with sulfate reducers. Also, they are interesting in terms of, like in the sea, they are very important because you have a lot of sulfate. So we are also looking to processes that involve sulfate reduction, but that's more on the environmental context and not so much on the biotechnology context.
However, I mean, if there are industries which emit a lot of sulfur kind of emissions as a syngas or a flue gas, I don't know what the right word is.
Yeah. So there's also even companies like Pax in the Netherlands that they work a lot on the desulfurization of these streams. So they do remove the sulfide from this gas, also using microbes.
Then you can produce elementary sulfur and reuse it. Yes.
Really?
Yeah, it's done on a very big, large scale.
But how does that work? I mean, they use the sulfate they digest that, or they just take things off and put things together, and then it is as a mineral dissolve in the bottom.
You get to produce the sulfur. You do a partial oxidation of your sulfide.
Okay.
And there are bacteria that can do that. So yeah.
So basically what they do is they bring energy or take energy, and then they take what they need and then dump the rest.
You shouldn't add too much oxygen because then you get sulfate, and you're sort of stuck with sulfate.
Sulfur is more valuable.
Sulfur is more valuable, more usable, but still not very valuable, I'm afraid. But
no.
So
but sulfate, I work with sulfate reducers, but more from the point also of one carbon metabolism. So we were studying different pathways for one carbon in sulfate reducers as well. So that we found, maybe this is too technical now or too microbiology detail now, but we are also interested in the group to find new pathways in microbes. So finding new ways of conversion, new enzymatic routes. And with one of the sulfate reducers we worked, the Sulfofundulus kuznetsovi, we were looking into methanol conversion by this organism. So methanol is also a C1 compound, and it can be used by many different types of microbes. It can be used by methanogens again to produce methane. It can be used by acetogens, so the ones that produce acetate, and sulfate reducers, and also, of course, by many aerobic organisms. And the pathway for conversion of methanol, it's well known in methanogens and acetogens, but in sulfate reducers there was not so much knowledge. And there's two main pathways known for this conversion anaerobically. So one involves methyltransferase, and the other one alcohol dehydrogenase. But they have been always detected like an organism has one or the other. And what we found in this organism is it actually has both. So this was the first organism found that had the two alternatives for the conversion of methanol. And that was quite nice because, it can also give the microbe flexibility in terms of the metabolism.
So then it doesn't need, probably doesn't need another syntrophic organism to do something.
No, this one can do it alone, yes. If you have sulfate, it does it alone.
Right. Because for me, the case of Lanzatech is a good example of how one microorganism eventually, and I think I think the last number I know was like 50. They found 3 metabolic pathways able to produce 50 kind of biochemicals, which is amazing.
Yeah, if you look to the portfolio of LanzaTech, of course, they have a lot of, they do, I have contacts with some, or I know some scientists in LanzaTech, and we of course go to same conferences, and because they are also very active in research, and I think that's a very nice thing from them, that they really invest also in doing research. So they do have a lot of nice research, and that they even make accessible in publications. But LanzaTech, they do have this portfolio, but they mainly focus on genetic engineering. So all these products, they are all produced by organisms that have their DNA added. So it's a bit different route than what we try to take here, because we don't modify some. We also have some projects, but more to get knowledge on how microbes work with synthetic engineering. But basically, we want to develop natural catalysts, because also of all the regulations and in terms of use of GMOs and for the robustness of the system. Because if you have more microbes together, then in general, you also have a much more robust system than when you have one microbe alone.
Yeah, true. And then they also exchange a DNA or RNA together, and then the whole DNA, the genetic pool also mixes together.
Well, that's also very interesting. That's something I would like to. I've always talking with this also with people in my group. Like, it would be really, because we have so many of these co-cultures now, and it would be really interesting to do more on the evolution of these co-cultures. So from the moment we put them together, and then years and years and years of cultivating them together, they must exchange and change something in their genomes, or at least they can, if we don't know. So it would be super interesting to. Yeah, so it would be, we still have to do that, because we really need to do that, because I think it's very interesting to see how these microbes, by being cultivated so long together, how they change.
You can sequence every week and then see how it changes.
Maybe we can do that together, Mark.
Yeah, we can do that. Yeah, sure.
Because I know that, for instance, plants genetically modify the bacteria inside the leaves and outside the leaves for their benefit. And if plants are able to do that, and the whole lessons that we learn from corona and viruses in general, I mean, humans are, Mark, I think what you mentioned, humans are 60% DNA that came from viruses from the past.
Yeah, you can find all kinds of virus-like parts in the genome of human beings, of course.
And that brings me to two interesting questions for the dialogue. Is one of them, what happens if you add viruses to your cultures?
Phages, bacteriophage.
Yeah, phages, just and see how, and see what kind of DNA changes happens. And two, it would, as mentioned, it would be really interesting to see if these microbes communities transform themselves just because they need to survive in a certain flue gas or syngas environment. And then who knows, we find the I don't know, the efficient community, and then we can put them in on every exhaust pipe of I don't know whatever industry. And then our syngas is being cleaned.
Yeah, well, no idea about phages. You actually read a little bit my mind, because when you were talking about using just one culture, and I was saying like organisms are more robust when they are together, I was thinking about phages, because sometimes when you have sometimes when you have a monoculture, and imagine you have a monoculture, you have an infection with a phage, and then the whole culture is gone. So if you have a mixed culture, at least the chances that some of them survive, because phages are quite specific, then it's higher, right? So that's one of the things with using mixed cultures, and maybe even now we work mainly with two, like I was saying, these two organisms, one making one reaction, the other one making other reaction. But there are also several that are able to make this first reaction, for example, the CO conversion. So we actually are running now some tests in the lab, mixing different types of organisms making the same conversion to see if they all stay in the culture or if they compete. But if they all stay, maybe it's even better, to create more diversity. Still, the reactions are controlled, but you create more diversity.
Yeah.
And this came a little bit also with the question we had with the pH, because there was a small mismatch in the pH that we have in the culture, and even the pH sometimes varies a little bit. So if you have, for example, microbes that are able to work at different pHs, and then you put them together, if you have small oscillations in our project, they will just, auto-regulate themselves. Yes, this is, I think, the type of flexibility you can get with these cultures, the mixed cultures.
And then you extract the gas or the ethanol out of there, and then you have another bioreactor with another group of bacteria turning that into something else, or? No,
we put them together in the same reactor. We mix the different organisms together.
Good.
Yeah, I think and we really showed also that's very beneficial, because you get a little bit that effect that I was explaining, the thermodynamics. It's very important for microbes because they gain energy from these reactions. And I was explaining that in the case of the natural syntrophs, it was very important to remove the hydrogen for that first organism to work well. So we see exactly the same thing also with this Clostridium co-culture that we had for CO conversion, because the second organism is taking the ethanol, that is one of the products from ethanol and acetate that are the products of the first reaction. So by keeping those products low, the thermodynamics of that conversion done by the first organism is also better. So in that sense, that organism also beneficiates a bit from having the second one.
Right.
But is it then a continuous reactor, or is it batches? Because somewhere you have to get the product out of the system.
Yes. So we've done batch or continuous. So you can, but we for many things. For proof of concepts, often we go for the batch because it's easier to screen and you can make different trials. But when we want to test the system, then we go for continuous in chemostats.
Yeah.
So a continuous process that you have inlet and the outlet of the liquid.
So what makes your work probably quite difficult is you want to use two strains only, or so two different types?
Not only, but at least two.
Yeah, at least two. But not everybody else who can also be there. So you have to work very on the very strict conditions, isn't it? To prevent any kind of
sterility of the system. We always say that's something still to test, because we always say okay, CO is quite toxic for many organisms. So probably there will not be a high risk of contamination. If you look also to the pure culture process by Lancetech, they also don't, it's a pure culture and a monoculture, and they also don't sterilize, and it runs well. And that's also because if you are running a glucose fermentation, I would not say the same, but with CO, it's also a good agent to kill maybe contaminants.
Okay.
But you're right. That, I mean, that's a risk, and it should be also more assessed. Like how sterile can you keep, or how free of unwanted microbes can you keep these systems?
Just a question, Bob. Can I? So actually, you maybe I can ask you a question from another perspective. I told you in the beginning that we are we're trying to find PFAS degrading organisms in nature. And quite recently, we've done a lot of sequencing on samples that are contaminated, groundwater samples contaminated with PFAS. And then we find a syntrophic organism that gets enriched when there's more PFAS. Would that suggest that there's probably also other ones that actually attack the PFAS, or?
Yeah, well, I think that
you don't know the details, but
no, but I think, and I think I'm not the only one thinking this. This is maybe common sense that much of the or many of the non-cultivable microbes now. So we cannot cultivate them in the lab because we are always giving the same conditions, but we also aim always to have pure cultures. And my view is that we should do it differently.
Right.
We should just try to isolate syntrophic organisms or combinations of organisms. And this, we have also an interesting work going on now that was not so much on. So we wanted to enrich for syntrophs. So we use these syntrophic substrates for the enrichment. Yes. And then this is a work that is in collaboration with a lab in China, and they did most of the experiments there. And by using these syntrophs, they enriched for a very for a methanogen that is in a very unknown branch of the tree of life.
Yes.
And I think that worked in that way because the target was more the syntrophy. So it was more like checking the substrates that were used initially already selected for microbes that were good at growing together. So there you already select for different microbes than you would select normally when you really try just to go for pure cultures. So I think that if we develop, and that's really something I think we should do, develop new ways of enriching communities or pairs or more bacteria that are able to work together. Maybe like that, we will be targeting more the new microbes and not just obtaining always the same that we do with the current ways of isolating.
There will be other ones out there that can do even better. Is it right? Yes. And you want to find those.
Yeah, that's another thing I really like, to find new microbes. And
Actually, you find one, wasn't it?
Well, in our lab, we found many, but indeed, during my PhD, I isolated or co-isolated a syntrophic bacteria that is able to grow on the long-chain fatty acids like oleate that is present in olive oil. So it was growing in oils, basically. That was very nice because these microbes are very difficult they are so dependent that sometimes with the techniques we have in the lab, we don't know enough about collaboration and we don't promote this collaboration. So it's, I think, more difficult to sometimes isolate these syntrophs. It's more difficult to isolate these syntrophs than isolate, fermenters that can grow on their own.
So, we found these desulfomonyle bacteria that are somehow related probably to PFAS. So let's hope that we find more of these bacteria in high PFAS concentrations, and maybe find other ones that work together to degrade the PFAS. Actually, I hope so. But could it also be that the syntrophs are enriched through some kind of a toxicity effect of the PFAS or something like that?
Or some of the intermediates in the degradation. What's the main intermediate there for degradation? What kind of?
There could be so many. It's so hard. I mean, it's a large molecule with all fluor on it.
Yeah. But when you remove the fluor, take
off one of the fluors, and then you have another compound. So it's also a lot of fluor on it, but one less.
Because this the sulfomonyle, I think they can also grow on. I even think that they can grow on fatty acids, for example. So if that's also present in the medium, then maybe they are just doing that. It can be,
It's the groundwater. It's the natural population that we're looking at. So, well, we still have to find out about this, but
that's even more interesting if it's done by more than one microbe. It's even more interesting than just one alone.
Probably
it's a complete army with all their own specialisms to attack a PFAS molecule.
Yeah.
Maybe. Why not? Seeing it as I normally say that for this, when I talk about co-culture, sometimes to explain, I also use the analogy of an orchestra. Like you can select the instruments and the players, and then as a maestro you make the music, and also you make the conversions you want to make. using this. But well, maybe you can also see it as an army.
It's wonderful. But I like that analogy a lot because I like music a lot. But how is it coordinated then?
You are the maestro, so then you have to coordinate. You are the composer, maybe, and the maestro, because, well, the composer, you compose the culture, and then the maestro, because you define the conditions and you define how they are interacting. So, yeah.
How is nature doing that?
What?
How is nature doing that?
Mark, I can imagine that you need to know the pH level, you need to know the minerals, trace elements.
Yeah. I know there are many bacterial species out there, but how do they coordinate with each other? Yeah, we already discussed a little bit about.
Yeah, nature is the best composer of all, of course. That we can unfortunately not yet completely understand. Otherwise, we could just copy.
Yes, we have to.
Yeah, very little, and I think we really need to dig into interactions to understand more, because I think we know. I would not say a lot, because if you look to the tree of life for microbes, then you see many things we don't know, but we know a little bit about physiology. But if you look up how much we know about interaction between microbes, that's even less. You can look to a very complicated system and try to figure out, but I think it's so complex that we also need to do more studies at increasing levels of complexity to really find out how microbes interact with each other in a more broad sense.
I agree so much.
In a normal way. And there was one research on your group which also found my interest. It's the molecule called ectoine.
Mm-hmm. Ectoine.
It's a 1,456 tetrahydromethyl penamyxyl. It's a kind of an acid found in several species of bacteria.
Yeah, especially bacteria that grow at high salinity, so high concentrations of salts, like in salt lakes, or they have this molecule. It helps them to survive this high salinity. So they produce it as a way of defense to protect themselves from the environment.
And then it's good for skincare and sun protection, but then it's then logic because it's.
Yeah, it's used a lot. I think it's because of this. It's also good for your skin, so you can incorporate it indeed in creams, or and there's also medicines, and it can be produced from sugar. So that there was research, and this was a postdoc, Sara Cantera. She came to our group because in Spain, in Valladolid, where they work specifically on ectoine production, they were looking on the conversion of sugars or other compounds to ectoine by this bacteria. But when she came here, she wanted to look also if it would be possible to convert CO2 to ectoine. So she's now doing research on that. So also with carbon monoxide and carbon dioxide and hydrogen, looking if this can also be substrates for these type of organisms or new microbes as well. So we looked also into the genomes. We found microbes that have the genes to produce this ectoine and also have the genes to produce to use CO and CO2 and hydrogen. And now we are testing them to see if they are able also to produce this compound from these one carbon compounds.
Yeah, which is a good thing, because sugar, we have a shortage on sugar, and we cannot put every sugar kind of molecule into fermentation. Then we would not have any food left for the rest of the species.
Yeah, that's true. That's why we try to look into substrates, conversion of substrates that are less essential for food, or that you can frame within the circularity that we want to achieve in the future.
And that's because sugar is sort of the easy metabolism, and if you move away from sugar, you go into uncharted areas, basically, from traditional research.
Yeah, sugar is a very good energy and carbon source for organisms. They can make a lot of energy from this molecule. So they prefer that, or some microbes, of course, prefer that. But that's the good thing about microbes. They live everywhere. And in some places, there's no sugar to convert. So they had to adapt to convert other things.
Something else. Right.
So we have to search for those and try to see if we can convert also other molecules.
And you also been working with bacteria usually at a pH level 2 and lower. I have never knew that there are bacteria surviving at pH levels lower than 2. I thought that was sort of the max of life.
Yeah, that's the main expertise from Irene Sanchez Andreas. She's also working, she's assistant professor in our group, and she really specializes in this low pH, and she has done a lot of research with samples from Tinto River in Spain. That's a natural ecosystem there that is also very low pH. So she isolated several of these very low pH or species that work at very low pH, which is very nice, and also has an application in biotechnology, because when you have extraction of metals in mines, you generate these mining waters that also have very low pH. And you can also use microbes to you can recover the metals in these wastewaters by using microbes. So these microbes are able to, for example, reduce sulfate and produce sulfide that we are also talking before. In this case, sulfide is the good one, because sulfide precipitates with the metals. And then you have a metal sulfide precipitate that sediments, and you can remove from the liquid water again, and in that way purify the water and recover the metal.
Yes, great. Metal precipitation.
Yeah.
There is nothing that bacteria cannot do, it seems.
No, maybe if you can think of a bacteria, something that a bacteria cannot do, maybe that's
there will be
that's
one
the challenge.
That's a difficult one. That's true.
Yeah, and there was also a collaboration with Shell on electron receptors.
Oh, well, with Shell, we have Shell and also some other companies. So we have some projects now on hydrogen subsurface storage.
Yeah.
I don't know if that's what you mean. So, of course, we move towards a more renewable economy, and we have to leave petrol behind, right? And a good energy vector is hydrogen, because you can produce hydrogen from aeolic or from solar. So there's many of these traditionally petrol companies that are looking into ways, what are we going to do after petrol? And one of the things is indeed then producing hydrogen from, because solar and wind, you can produce, well, solar during the day, but there's peaks, right? So it's not when you need, it's normally not when you produce it. So a way to have an intermediate storage of this energy is by producing hydrogen. But then still you need to store this hydrogen to use it when you need. So the idea is to have a way to store the energy that is produced from solar or wind. So hydrogen, then it's the better. Now, where do you store hydrogen? If you would have, for the needs we have of energy, if you would have to store energy just in hydrogen just in tanks, we don't have enough area and enough volume to. You would have to have huge parks with tanks to store all the hydrogen. Yeah. So these petrol companies, they have empty reservoirs, like oil reservoirs that were, they extracted all the oil, so now they are empty, or caverns where they also used to store, for example, methane, that now are empty. So they want to look if these places that are underground and they have very big volume, they want to see if they can use these places to store hydrogen. But when you store hydrogen there, hydrogen is a very good energy source for microbes.
Yeah.
So maybe
you
when
get
you store hydrogen there will be some conversions.
Yes.
So we are looking into that. We now get different samples from different places, from different countries, from these oil reservoirs and caverns, and we incubate them with hydrogen, and we see what happens to hydrogen. Is it converted or not? So can we store hydrogen in these places or not?
But then again, if you combine hydrogen or syngas in the storage, including the microbes, then you have a natural conversion factory again, either back to methane or I don't know which direction, and you just can pump up your product again.
Yeah, there's also that possibility that use these big storages as bioreactors, so that you can produce again methane to refuel so you inject hydrogen. In that case, you have to have hydrogen and CO2 as well. So with hydrogen and CO2, microbes can make methane. So then you would be able to make methane and to refuel naturally
and most likely you need to add more bacteria to it just to make sure the reaction gets going.
Probably, yeah.
But then,
that's something we will also find out. Like we have some incubations, and we already find differences also from the different sources. You have faster or no conversion at all of the hydrogen. So it depends also on the site.
Yeah, because everybody's saying we're going to store syngas back into the old gas fields. So I'm saying that is so stupid. If you do it without microbes, that is so stupid. Add microbes to it, and then they will do their magic, and then you suddenly can pump a product. Now it's a waste stream, but it's feedstock for bacteria, and then you just can deplete the reservoir again with just deriving a new product out of it. But that is when you understand a little bit of microbes, you would think, that is just logic. Just combine things. Don't go mono.
Yeah, well, the good thing of this process is there's really transdisciplinary type of a team, and we are looking to microbiology, but there's groups looking to the chemistry, geochemistry, everything. It's being looked at. So that's very nice. Let's see what happens there. But it's not just the Netherlands. It's something you see in Europe. So this is a very a direction people are exploring.
I would love to have some names of people in that area just for the interview, because it's generic. The narrative is like, we just dump it, and then hopefully this CO2 doesn't come out of the ocean through holes. But if you add microbes to it, you have a bioreactor. Just use the bioreactor, which is, was it 6 million cubic meter? Okay, let the stuff do what it does. I mean, we've all even find microbes. Was it 2-3 kilometers deep in our soil, and nobody thought that it would actually be possible that something is living there.
No. And one thing is what is living there now, and another thing is, of course, if you give them substrates in which they can grow, they will grow. So you will have also development. It will change. So the microbial diversity in these places will likely change. If you start giving them a substrate, they will start growing. You'll have new communities being formed, new intermediates. So you create a food chain also in these environments. So that will also change.
How cool is that?
Yeah.
Well, I think it's very nice, but let's see how it develops now.
Yeah, interesting stuff. Yeah.
It's not easy for the generic industry to think about the beneficial effects of microbes in systems.
What's your sort of experience? Is there enough open-mindedness in the outside world for microbiology? What do you think?
Well, I think that's dependent. I think we are in different phases of knowledge, maybe like chemistry. I think it's more controllable, so people trust it, like it's reliable. It always works in the same way if you have certain conditions. And microbes, they evolve and they interact, and they are a little bit more unpredictable.
They're like humans.
Yeah, but if you get to know them better, then I think this will also decrease. But I think we are just at different stages of knowledge. And I hope that in the future it becomes, indeed, there's still some resistance from industry to really go for microbial processes. But I hope that changes. And one advantage is that microbes they are very sustainable. They produce themselves, right? We don't have, it's not like a metal catalyst that you need to have the metal and then you need to regenerate or you lose even the activity. It's like microbes, they grow, so they produce the enzymes that are the catalysts themselves. So that's a big advantage.
It's a big improvement, and they use less energy and less resources. And
to elaborate on your question, Mark, for you, Diana, what is the impact LanzaTech is having on this space? Because they are doing something completely against chemistry. And still
I think a big impact. And I really like the history of LanzaTech because it started with an idea from a person or a few persons, I don't know, but it started with a very crazy idea. This organism that they started with was not that fast or good in using carbon monoxide, clostridium autoethanogen, we still use it. But they just believed in idea. I think in the beginning, once I heard the talk, and then they were saying like, we started in a garage just doing the experiments. So I think it was like, we trust on this and we are going to make it work. And they did. And that's remarkable that they did it in the time they did, because I think they started around 2000. And I think in 2010, they had it shown that it was possible and they really grew. And now they are an example. And I think a good example that it worked, because there's also many bad examples or examples that did not work. Maybe not just because microbes were not good, but maybe there was not enough persistent, or like LanzaTech is really the example of starting with an idea, you believe on it, even if others don't believe, and then you work on it, and then it works, and then you show to the world that you can do it, and you can have different alternatives for processes. So in that sense, I really think it's a good example, and I think it's helping the all. I think we should. There's still room for many more LanzaTechs, let's say.
Right.
So that's what I hope, that other companies also start developing and applying different technologies using microbes.
Yes, wonderful. Well, let's work on that.
Yeah.
And what is your vision then on science? Because I've read some of your notes on Twitter, and I know you have a certain opinion on where science should be heading towards.
Well, that's I don't know exactly what kind of ideas I expressed on Twitter now, but I think science at universities still should be basically fundamental, and this doesn't mean that we are losing the application side, because I think it's very nice to have in view some applications, but I think it's also the place to really explore and find new things. And in that sense, I think there should be more support for this type of research. I see that nowadays nowadays when you apply for a project, many times you already have to have results and you have to know that it's going to work. But what's the risk there? It's I think there should be also more incentives. Just it's a crazy idea. Maybe 99% of the cases will fail. But that's also like in pharmacy, they also invest huge amounts of money, and then for a positive, and then 1 in 100 they get a very good result. And I think science is like that. You cannot guarantee that you'll get always.
Penicillin was an accident. So. Science runs on accidents, and accidents suddenly turn into something useful because suddenly you look at it from a different perspective. And
your eyes open, be creative. Yes. Yeah. You see things and you take it further. Yeah.
Yeah. It's being curious and it's being observative. and not just looking into what you want to see. I think that's one of the things, because many times the interesting things are in the things you don't want to see. So you expect to get that, but many times the interesting thing is in something else. And if you are so focused on just looking into what you want, you just miss the interesting aspect.
You miss your Nobel
and also
Prize.
the way we do research nowadays is too much based on output. So I really enjoy reading old papers, because old papers you see, like I'm sometimes like, how did people in the 70s do all this nice research and so much knowledge is in one paper? Because I think people were just, they had an idea, an hypothesis, they were testing. There was like a very comprehensive research, and then there was the publication, but it was not publishing for publishing. It was, and nowadays there's a lot of pressure for publications, and sometimes the work that is not adding that much. So there's you read a publication and you take a small message, but I think we should also really, who even reads all these papers? There are so many papers that we cannot keep up with reading all these papers.
Only make a paper when you have something to tell, isn't it?
Yes, and not just because we should. I mean,
yeah.
So maybe I don't know if these were divisions you were
no, but
referring to, but
yeah, sort of, and it's perfect in line with the people we have in this podcast. Everybody tends to be a little bit odd and curious, and more of a designer or a creator than actually a scientist, or even they are a scientist. I mean, art and design and science is so closely together. And we tend to invite those who have an open view on things, and that's the fun part with conversations, that you can go in all directions, because we like to explore different angles, and then sometimes we have, we never thought about that, and suddenly it's like, oh yeah, maybe who knows.
Yeah, well, that's very nice. I also like combining art and science, and there was also in the 100 years of Wageningen, there was an artist in residence at Wageningen each month. There was an artist in residence, and I was together with one of them. And then he built also, he built a synthetic co-culture, a synthetic culture of microbes. So it was an electronic circuit with different, so a circuit connecting different chips, and then they would lighten, and the light would be influenced by the ones near each other. So they were kind of communicating and creating a pattern in lightning. So that was also very nice to show how communities and interactions in microbes work on the art with the art perspective.
Very inspiring, yeah.
And then we're very close to the microbial fuel cell again.
Yeah.
Okay.
I think it's let's wrap up.
Yeah.
Okay. Any last question, Mark?
Oh well, so many questions. I would love to talk to you more about syntrophs and whatever, but we will find an opportunity to do that. I'm quite sure.
I'm sure, and you are also welcome to pass by in Wageningen.
Yeah, I will. Yes.
Definitely will. It's around the corner. Nowadays we can travel again, so luckily.
Yeah, we're free again. Yeah.
Yeah, we're free again.
We don't know for how long, but
that's true. That's why we have to take the opportunity.
Exactly.
No, great. Thank you, Diana, for having you on the show. And,
thank you so much. It was really fun to do this. My first time, but yeah, I really enjoyed it. Thank you.
You're welcome. And for all the listeners, see you all until next time on the podcast. Ciao. Bye-bye.
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 I wish you enjoyed this episode of the Master of Beautiful Achievements podcast. Do consider joining our Patreon community via the link below to help us make and record more of these episodes. If you like our podcast, subscribe in your favorite podcast app and do leave us a review to increase our ratings. And if you want to know more, check out the Masters of Beautiful Achievements website. A special thanks to our sponsors Scope Matters and Orvion for making this series possible. Thank you for listening and join us next time.